Hello, everybody. Welcome to episode 11 of the Living Environment Podcast. In this video podcast, we'll be looking at the controlled experiment, how to run a scientific experiment, and what types of questions you'll see on the Regents. Okay. When you design an experiment, here are kind of the main factors you have to think about. The first one is the problem. You'll sort of have some problem that you'll want to solve. 2 is the hypothesis. 3 is your setup and what variables go into that setup. 4 is what data you'll be collecting. 5 is a conclusion, whether your hypothesis was right or wrong. And 6, you have to decide if your experiment is valid. So let me go over those. Let's start with the problem. Got a problem? Yes. It's what you're trying to find out. So the Regents will usually write a little paragraph about what someone is trying to discover. Like, for example, one year, the Regents question was something about how fast wounds heal. And so what they are surgical wounds. So someone's in the hospital, and they wanted to find out if they took vitamin C, would the wounds heal faster or not? So that was the problem. Okay, and they'll write a little paragraph to introduce the problem to you. Now, here's another example. If you wanted to find something out like this, does extra sleep help students perform better on tests? It's a problem that you're trying to find out. That is not a hypothesis. A hypothesis is a prediction that you make. It's sort of a statement. The one thing it can't be is in question form. So here's some examples. If students get 2 extra hours of sleep, test scores will increase. Number 2, increased sleep will result in increased test scores. Number 3, students that sleep an extra 2 hours will do 10% better on tests. Okay, these are all possible hypotheses. The only thing you have to make sure that you notice is that they're all in statement form. Now, one thing you should not do is start with the I believe or I think. It's your hypothesis, so you want to make a determined statement as opposed to sort of like, I think this will happen. 3, the setup. All right, this is your experiment. You want to set up 2 or more groups. Now, one group is called the control group. The control group is the normal setup, the one that is sort of the one you compare your results to. So, for example, if you're doing an experiment on plants and trying to see if, I don't know, milk and Sprite and soda, if it affects the plant growth, then the control group will be a plant that just gets water. It's a normal, very typical setup. That's your control group. So let's do the test grade, the test score. We're going to get 2 groups of the students. And for this test experiment, I want to get the same group. Okay, the control group is a large group of normal that receive normal amounts of sleep, and then they'll take a test. And the experimental group is a large group that gets extra sleep and then takes the test. Okay, so this is the difference in this experiment. Control group, experimental group. Now, when you do an experiment like this, if you notice, you need a big sample size. Sample size is, instead of doing the experiment with 2 people, which is really dumb, you want a large average just to make your results better. So you wouldn't do the experiment with just a few subjects. You would take a large sample size. The placebo, also known as the placebo effect. This is done, a placebo is used in drug studies. If you have a control group, you have to give them something. You can't just take, if you're doing a drug study, half the people and give them nothing. The person has to think they're getting something. And so a placebo is a fake or sugar pill that the person takes. That way your results won't be skewed by people knowing they're not getting the medicine. Sometimes human behavior and the brain is weird. So you try to fool your subjects by giving them a fake pill. And that fake pill is called the placebo. All right, when you set up your experiment, you actually control everything. They're called the variables, the factors in an experiment. So the whole point is everything is the same in both groups except one thing. So if you were doing the plant growth, everything is the same amount of sunlight, the same position in the room, the same type of pot and soil. But the one thing that's different is one gets water and the other one gets soda, milk, or whatever. So the variable that's different in our test taking one is amount of sleep. And the variable that must stay the same, well, pretty much anything. The age group of the people, the subject they're taking, the time of day they take the test. It could be anything, as long as it's not how much sleep they get. Number 4, your data. The Regents likes this, so be ready. They like to ask you what data must be collected for this experiment. So it's data that's measurable, usually numbers, and it's going to support or refute, that means not support, your hypothesis. Now here's the number one incorrect answer we get on the Regents. The Regents will ask something like, give an example of data that you're collecting, and someone writes stuff like this: the amount people slept, or how long people slept. Wrong. Or how much medicine the patient took. Wrong. Or how much growth solution you gave the plant. Wrong. That's not data you collect. This is stuff you measure for your experiment. Data you collect is like this: the scores people get on math tests, or the amount of time it took the wounds to heal, or how fast the plant grows, or growth rate of the plant. Those are correct data answers. Conclusion or example of results. Sometimes the Regents will ask you to give like fake data, or they just ask for, please give an example of results that support the hypothesis. Or sometimes they are wacky and say, give an example of results that refute the experiment, say that it's false. So give example of results. So here's mine. Students that slept an extra 2 hours did 20% better on tests. That's totally, we don't know. We didn't do the experiment. We just made it up. Here's another one. In 2 weeks, the plant with the growth solution grew 12 inches, while the plant with water only grew 7 inches. Okay? That's an example of results. 6, is the experiment valid? This just means, and this does come up on the Regents where they show you a poor experiment and they ask you what's wrong with it. A lot of things that are wrong, like, is if the person, for one, uses a small sample size. 2, they have too many variables that are different. Those are bad experiment questions. This is a little different. This is, if the experiment is valid, means you ran a good experiment, but no one's going to believe you unless 2 things happen. The validity of an experiment, how true it is accepted if, 1, the experiment is repeated many times with equal results. That means you do it over and over and over, and the same thing happens. And 2, you don't do it. Let other scientists do the experiment, and they get the same results. That's a good experiment. So let me show you 2 Regents questions really quick. This one said, design an experiment consisting of a control group and 3 different experimental groups to test the prediction: garlic grows better as the salt concentration of the solution in which it is grown increases. So basically they're saying the saltier it gets, the better garlic grows. And then it says in your answer, be sure to. Now remember, we said you don't have to write a whole paragraph. You can actually answer the bullets. So let's do bullet by bullet. The first bullet: describe the control to be used in this experiment. Now, you could say garlic, it says as salt concentration increases. It doesn't say garlic grown in salt. So the control group in this experiment should be garlic grown in, let's pretend, 5% salt concentration. And then the second bullet says, describe the difference between the 3 experimental groups. Well, I'm going to say experimental group 1 is 10% salt, experimental group 2 is 20% salt, and experimental 3 is 30% salt. So I made my control group be salty and the other 3 be saltier. You can say the control group is freshwater, but I would stay away from that. It says as salt concentration increases, but I'm pretty sure that freshwater is probably accepted. The third bullet says one type of measurement that should be made if your prediction is accurate. That's a fancy way of saying collecting data, okay? So the measurement, what data you're collecting, is growth rate of the garlic, or how fast the garlic grows. And one example of results is the last bullet. Well, the garlic that grew in the 30% salt grew 10 times as fast. Something like that. And this other one, this was another Regents question. Describe some parts of an experiment to determine the preference of pill bugs for light or darkness. In your answer, be sure to state a hypothesis. Now, if you notice in the last Regents question, they gave you the hypothesis. In this one, you have to come up with it. So you just make something up. You either pick light or dark. So I'm going to say pill bugs will prefer the darkness. Beep. Second bullet, identify the independent variable. The independent variable is the one in the bottom, if you remember one of our other episodes. So in this thing, it's the amount of light. So that's what you're trying to test. You're trying to test, as the amount of light changes, what will happen to the pill bug preference. 3, identify 2 conditions that must be kept the same. Now notice there's a number 1 after this bullet. It did ask you for 2 things. Usually you see a 2 after when they ask you for 2 things, but here there's a 1. What that means is, you actually have to get both right to get a 1. Okay, so 2 conditions that must be kept the same is anything. Anything except light or darkness. So you can say the species of pill bugs must be the same. The age of the pill bugs, the temperature of the room, stuff like that. And the last one, again, one example of experimental data. Pill bugs preferred the darkness, and it says support your hypothesis. So my hypothesis was about darkness. So I say the pill bug went totally to the dark like 19 times and went to the light only 4 times on average, something like that. And that's pretty much it. Hope you run an awesome experiment. Know that the Regents lately has been doing shorter experimental questions. They seem to be going for more short answer. But again, we cannot predict the future. So be ready for any kind of experimental design questions. Okay? Good luck.